I'm really pleased to introduce our Biochemistry and Biophysics capstone class, who selected stress and resilience as a life-relevant topic to understand at the molecular level. We discovered that it is both super complex and impossibly broad. PubMed lists over 250,000 papers just with the word stress in the title. Though we are far from experts, we've learned a lot of fascinating things, a few of which we can share with you today.
Stress. It's something that we've all experienced. College students endure many stressors like moving away from home, living in a small room with multiple strangers, and keeping up with rigorous academic expectations. So that's why this topic was of such interest to us. According to a 2021 Gallup poll, around a third of undergraduates had considered withdrawing within the last 6 months. 76% of those reporting emotional stress as the main reason, with other common reasons like the pandemic, cost of attendance, and academic difficulties also being stress-related. Furthermore, according to the American Addiction Centers, common coping mechanisms include what might be considered more healthy ones like physical exercise, meditation, and therapy, as well as less healthy ones like eating, consuming alcohol, and using study drugs. And clearly, stress impacts not just college students, but everyone.
In fact, the global stress management market has been estimated to be about $20 billion. So let's look at how we will cover this topic. Today, we will first cover some key concepts and biochemical players related to stress and resilience, then some fascinating biochemical details, and finally, on the more practical side, we will look at some interventions to combat stress. And as a strategy to ensure that we are covering topics of importance, we purposely focus throughout on work in highly cited papers, what we call landmark papers. We hope that each of you will come away with a better understanding of stress and how to reduce it, as well as an appreciation for how the biochemical events involved are even more complex and remarkable than we might have imagined. So let's get started with some key concepts and players.
Our first key concept is from a simple 1908 study by Yerkes and Dodson. In short, they gave rats a choice between a black box that delivers a shock and a white box that does not. They hypothesized that the stronger the shock, the quicker the rats would learn to avoid the shock box. To their surprise, as shown here, there was a sweet spot, with increasing shock strength first stimulating learning and then hindering it, and with the hindering occurring earlier for the harder task. Out of this simple beginning, as described in this 1994 paper, the concept has been very broadly generalized in graphs like this one for how any type of performance depends on virtually any kind of stress. Next, let's look at the original focus of stress research. In 1936, Hans Selye published that organisms respond similarly to non-lethal severe damage from a wide variety of agents, such as exposure to cold, excessive exercise, or surgical injury. In this view, stress was virtually any challenge that happened to an organism, and that induced a common response or syndrome that has 3 physiological stages. First is the alarm stage, where resistance goes lower than the baseline. Then comes the resistance stage, with elevated resistance to the stressor. And lastly, if the stress continues, comes the exhaustion stage, where the animal succumbs to the stress. In contrast to this externally focused conception of physical stress is the concept of psychological stress, which is what we are talking about today.
In 1984, Susan Folkman and Richard Lazarus produced a very influential book emphasizing that it isn't just what we face, but how we perceive a possible stressor that matters. We first make an appraisal whether the circumstance is a challenge or a threat. When we consider something a challenge, we experience less or even no stress. If we appraise the situation as a threat, in a secondary appraisal, we consider if we have the resources we need to cope with the threat. If we feel we have the resources to handle the situation, we tend to engage rather than avoid the stressor. It sounds complicated, but we're doing this subconsciously all the time. To study these things, researchers need ways to induce psychological stress and also quantify the level of perceived stress experienced. When working with human subjects, the go-to method is the Trier Social Stress Test. In the TSST, participants are given 10 minutes to prepare a speech advertising themselves to some staff managers, and they're told that their performance will be recorded and scrutinized by experts. They're given paper and pencil to prepare their speeches, but their notes are taken away immediately before their speech, and they're prodded to continue speaking if they stop before 5 minutes. This grilling experience is pretty effective, as indicated by elevated heart rate, cortisol, and other common metrics of stress. To induce stress in rodents, As viewed in this article, scientists use simpler methods like forced swimming, exposure to bright lights, or, as shown here, restricting their movement for certain periods. Perhaps this approach reminds you of some of our stress-inducing workplace environments. So, how are stress levels and resilience quantified? The tools are remarkably simple. For determining someone's level of stress, the perceived stress scale, created in 1983 by Sheldon Cohen, seems to be the standard in the field. It's a 14-point questionnaire that asks on a scale of 1 to 5 how often in the past month participants felt a certain way, such as overwhelmed or confident. And for quantifying a person's resilience, the tool of choice seems to be the Brief Resilience Scale from 2008, which is a 6-point questionnaire with questions asking people's sense of their ability to bounce back from stressful situations. As the last of our general concepts to cover, we want to touch on the importance of recognizing that societal inequities shape the challenges each person faces and how they cope, as well as how stress research is done and presented.
In this regard, biological sex and gender, race, and socioeconomic status are obvious huge factors to consider. As one example of how these factors are linked in the US, I show here how the median income of women is much less than that of men, and how it also varies with race. For this reason, women in minoritized groups may experience both an increased exposure to stressful events and have fewer resources for coping. Finally, as you may know, another factor is that historically, both researchers designing stress studies and the main subjects of studies have been Caucasian males. A couple recent reviews of this topic are listed here, and in the next slide, I'll share a well-cited study on a remarkable example of race-associated differences. This study correlated stressors, unhealthy coping behaviors, and depression by race. As you might expect, for whites, the number of unhealthy behaviors a person showed correlated quite strongly and positively with their level of depression. In contrast, while African Americans also had a strong correlation between unhealthy behaviors and depression, it was in the opposite direction, with higher depression seen among those who had engaged in none of the unhealthy behaviors. While we don't have time today to discuss what is going on here, the study makes very clear how important it is to consider external factors such as race and socioeconomic status when developing treatment plans and attempting to treat stress-related issues in humans. Next, we will introduce some of the key molecules involved in the stress response.
Our psychological assessment of and our neurophysiological response to stressors is hugely complex. Stress involves bidirectional communication between multiple parts of the brain and reaches into every body system. Conditional responses are guided by sophisticated positive and negative feedback loops across systems. Topics covered today only scratch the surface of stress, but we found them to be foundationally informative. Now, let's introduce a couple of key stress hormones.
There are 3 key hormones associated with the stress response. Each of them is released in response to a stressful event when triggered by the central nervous system. Epinephrine and norepinephrine are water-soluble tyrosine derivatives that are released from the adrenal medulla when triggered by the sympathetic nervous system through neurons in the spine. Cortisol, on the other hand, is a fat-soluble steroid hormone released from the adrenal cortex when triggered by the HPA axis. The water-soluble hormones diffuse easily through the blood and tend to trigger a cytoplasmic response via membrane receptors. The lipid-soluble ones, on the other hand, must be transported by carrier proteins, diffuse through the cell membrane, and usually alter gene expression in the nucleus, which is a much slower process. As you can see here, monoamines and peptides, which are generally considered water-soluble, take effect within just seconds to a couple of minutes, while the effects of lipid-soluble corticosteroids take on the order of hours. Now, let's take a closer look at epinephrine.
Epinephrine, the first hormone isolated, was sought after around 1900 as a molecule from the adrenal glands that raises blood pressure. John Abel published first and named it epinephrine, but actually had a benzyl ester of it. Jajki Takamine isolated it and named it adrenaline, which was patented by Park, Davis and Company. Note the 2 names are just Greek and Latin versions of the same thing. In Walter Cannon's 1915 landmark paper, Bodily Changes in Pain, Hunger, Fear, and Rage, he proposed epinephrine's central role as a hormone in the fight or flight response. In biochemical pathways, epinephrine signals through a G protein coupled receptor, and one of its effects promotes energy mobilization via the secondary messenger cyclic AMP, as shown here. but as that is well covered in general biochemistry, we won't spend more time on it today. Instead, we'll turn to the other key stress hormone, cortisol.
You won't live very long without cortisol. Glucocorticoids, cortisol in humans, corticosterone in rodents, are often described as bad actors. However, they are part of multiple biological functions in daily life and key to regulating homeostasis. Discovery of glucocorticoid importance began in the 19th century when Thomas Addison reported that adrenal extracts improve chronic fatigue, muscular degeneration, and other symptoms in patients. Researchers in the 40s found that treatment with isolated hormones from adrenal tissue alleviated arthritis and other conditions. Despite early discovery of anti-inflammatory action, the name glucocorticoid is a portmanteau derived from their action in maintaining fasted blood glucose. Mineralocorticoids, also important in stress, are similar molecules produced in the adrenal cortex, but involve sodium and potassium transport. Glucocorticoid function in a number of roles besides inflammation and glucose metabolism, including embryonic development. Glucocorticoid receptor knockout rodents die early in development because of improper lung formation. Also, glucocorticoids have roles in eye health, cardiovascular regulation, and important memory formation and recall functions. A great illustration of cortisol involvement in daily life is our diurnal cycle. Cortisol starts rising before we wake and gradually drops to a low during sleep. This cycle shifts to match changing sleep-wake cycles. Much needs to be uncovered, but researchers think that cortisol helps our body prepare for the day ahead, both in terms of energy metabolism, modulation, and consciousness level changes. These changes include boosting motivation and aiding a brain reboot after sleep, allowing retrieval of key memories. But how do our bodies access or create cortisol and other corticosteroids?
Cortisol is the main cortical stress hormone in humans. It isn't that way in mice and rats, the most commonly used animal models for stress studies. As it turns out, the gene CYP17A1 is silenced in the adrenal glands of rodents, preventing expression of the 17-alpha hydroxylase enzyme needed to produce cortisol. So they use corticosterone. One surprise was learning about a nomenclature fiasco in the field. While there is cortisol, corticotropin, cortistatin, and cortisone, the term CORT gets used a lot, which is not an acronym and is rather ambiguous. For instance, in a 2016 issue of the journal Endocrinology, some articles use CORT for corticosterone, as seen here, while others use CORT for cortistatin, as seen here. One last player to introduce is the nuclear receptor family of ligand-dependent transcription factors, as these are key molecules through which steroid hormones signal.
One subgroup of this superfamily are the steroid receptors, including the glucocorticoid receptor, or GR, and the mineralocorticoid receptor, or MR. Their basic structure includes an intrinsically disordered N-terminal domain that is a target for post-translational modification that modulates DNA binding specificity, a DNA binding domain, a hinge region, and the ligand binding domain. The ligand binding domain structure shown here varies to specifically bind different ligands, but the DNA binding domain has a highly conserved mode of DNA binding. These proteins originate in the cytosol, and ligand binding causes a conformational change leading it to dimerize. Then the receptor is transported to the nucleus, where it acts as a transcription factor by directly binding to DNA. The DNA binding domain is a zinc finger motif in which zinc ions help the protein fold. Specific side chains from the alpha helix, like the lysine, valine, and arginine shown here, reach into the major groove to recognize a specific DNA response element. Since the receptor is a homodimer, it binds to a specific palindromic response element. The specific response element for the glucocorticoid receptor is AGGACA with a 3 base pair spacer. The glucocorticoid receptor has the ability to act upon thousands of genes in carrying out its non-stress and stress-related functions. Now, with the key players introduced, we'll move on to part 2 of the talk, looking at some biochemical details of the stress response. We'll start at the beginning, in the brain, with the perception and appraisal of a possible threat.
As a very simplified overview, the prefrontal cortex, hippocampus, and amygdala are the brain regions primarily responsible for activation and extinction of the stress response. Key to an organism's survival is the ability to rapidly recognize and adapt to a threatening situation. The amygdala fills this role as our brain's center for experiencing fear and anxiety. Upon perception of a threat, it stimulates the HPA axis to release hormones which initiate the stress response. In contrast, the prefrontal cortex and hippocampus both tend to dampen the stress response. The prefrontal cortex provides cognitive appraisal and can counter signals from the amygdala. As for the hippocampus, although primarily involved in memory formation, it is also involved in mood regulation due to its deep links to the amygdala. Let's now look at some remarkable landmark studies that make clear the amygdala's central role as a primary fear center.
Understanding the role of the amygdala in people has been advanced by subjects with missing or damaged amygdala. For instance, patient SM has a disease that has led her to be missing most of her amygdala, as seen in the circled region of patient SM's brain. In 1994, a study was done in order to see if patient SM could perceive fear in other people's facial expressions, and she was unsuccessful. In a follow-up study, 9 patients with damaged amygdalas were asked to rate the intensity of 39 expressive faces on a level from 0 to 5 of happiness, surprise, fear, anger, disgust, and sadness. From this graph, you can see that the patients with amygdala damage poorly recognize fearful, angry, and disgusted expressions compared to the happy, sad, and surprised expressions. Control subjects with healthy brains and with brain damage in different regions were much more accurately perceived all of the emotions. In 2010, researchers subjected patient SM to a fear-inducing situation such as live snakes and spiders, haunted house, and a horror film, and not once during the study did patient SM exhibit a fearful response. Then, in 2013, a fascinating study was done on patient SM as well as 2 others with bilateral amygdala damage and found that they did have fear and panic response to air hunger from inhaling large amounts of CO2. This is the first time patient SM had experienced fear since childhood and implies an intriguing difference between external and internal sources of fear.
Neurons which release GABA-aminobutyric acid, or GABA, are referred to as GABAergic and are inhibitory because the GABA receptor is a ligand-gated chloride channel. When GABA binds to the postsynaptic neuron, it is hyperpolarized by an influx of negatively charged chloride ions, thus suppressing its ability to generate an action potential. Known for being the primary anxiolytic vector in benzodiazepines, GABA receptors in the amygdala and hypothalamus are particularly important for controlling levels of fear, stress, fear, and anxiety. One 2012 study sought to confirm GABA's significance in diminishing stress and fear by carrying out an amygdala-specific knockdown of the key GABA synthesizing enzyme, glutamic acid decarboxylase 67, or GAD67. They found that mice with the knockdown showed no difference in acquiring a new condition, fear, but were much slower to lose it, shown here, which demonstrates the role of GABAergic neurons in the natural dampening of stress and fear responses. Indeed, this figure from a recent review article outlines how the stress-terminating influence of the prefrontal cortex is accomplished by stimulating the release of GABA in both the amygdala and the paraventricular nucleus of the hypothalamus. This suppresses the amygdala's stress-activating influence while also shutting off the stress machinery itself.
The HPA axis is the name of the neuroendocrine pathway activated by perceived threats and signals the release of cortisol. HPA stands for hypothalamus, pituitary gland, and adrenal glands. The first hormone of the HPA axis is CRH, which stands for corticotropin-releasing hormone. It is produced in the paraventricular nucleus of the hypothalamus. CRH is delivered to the anterior pituitary and triggers the release of ACTH, or adrenocorticotropic hormone, into the bloodstream. ACTH is detected by receptors in the adrenal gland, stimulating the production and release of cortisol. Cortisol then travels through the bloodstream to tissues where it binds to glucocorticoid and mineralocorticoid receptors throughout the body, continuing the stress response. One key activity is binding to glucocorticoid receptors in the hypothalamus and pituitary to inhibit the release of CRH and ACTH. This negative feedback loop is crucial for maintaining homeostasis. We will now look more closely at just a few things in this pathway: CRH action, how cortisol synthesis is triggered and distributed throughout the body, and some aspects of the glucocorticoid receptor. Corticotropin-releasing hormone, the first hormone of the HPA axis, has 2 different receptor types. The first type binds CRH tightly and is responsible for triggering the HPA axis and releasing ACTH. The function of the second receptor type is less well understood. 2 different studies of mice lacking the type 2 receptor were conducted. The first study showed that the CRH2 deficient mice had a hypersensitive stress response, with ACTH levels peaking much earlier and cortisone levels also rising much earlier. In the second study, the receptor 2 deficient mice displayed comparable levels of anxiety-like behaviors at first. However, after 48 hours, they showed greater anxiety-like behaviors in every category tested. The current hypothesis is that CRH type 2 receptor is responsible for long-term stress adaptation, particularly in conditions such as depression. Next, we are going to be examining the final product of the HPA axis, cortisol, and how it is synthesized by the body.
So in order for cortisol to be released in response to a signal coming from the HPA axis, its precursor, cholesterol, must be transported to the inner mitochondrial membrane, where the enzymes that can convert cholesterol to pregnenolone and then to steroid hormones are located. This transport is mediated by steroidogenic acute regulatory protein, known as STAR or STAR-D1. STAR-D1 was discovered in 1994, and its centrality was proven by experiments showing that its expression, even in the absence of hormone signaling, promotes steroid production. How this protein works is pretty involved. First, ACTH signaling activates protein kinase A, which promotes de novo transcription of the gene and processing of the mRNA. The mRNA transcript is then bound to the outer mitochondrial membrane, where STAR-D1 is made and then phosphorylated. Next, STAR-D1 mediates the transport of cholesterol to the inner mitochondrial membrane, which appears to occur simultaneously with the movement of STAR-D1 itself across the outer membrane and into the mitochondria. Each copy of STAR-D1 moves about 400 cholesterol molecules to the inner mitochondrial membrane, and only newly synthesized STAR-D1 is active in facilitating this process. Once it reaches the mitochondrial matrix, STAR-D1 is degraded. The imported cholesterol is then processed into various hormones, including cortisol, which is released into the bloodstream for transport to the rest of the body. In the bloodstream, about 90% of cortisol is transported via corticosteroid binding globulin, also known as transcordin, shown here. The yellow cortisol molecule is highlighted with a black box, and it is bound within a hydrophobic pocket. At target locations, often sites of inflammation, a specific neutrophil elastase cleaves a loop of transcordin, causing huge conformational changes. These changes release cortisol, which can then freely diffuse across cell membranes. In cells, cortisol's availability is determined by the balance of active cortisol with its 11' hydroxyl group in red and inactive cortisone with the oxidized 11' keto group. This interconversion is catalyzed by 2 isozymes, 11B hydroxysteroid dehydrogenase types 1 and 2, each having the equilibrium shifted in one direction due to their coenzyme specificity. The type 1 dehydrogenase is in most tissues and is NADPH dependent, while the type 2 enzyme is NAD+ dependent and is only found in a few tissues, such as the kidney, colon, and salivary glands. In addition to the NADP versus NAD specificity difference that shifts the equilibrium, the Km values show that each enzyme has a 10-fold or more higher affinity for the substrate in its physiologically preferred direction. For its most studied functions, the next step is for cortisol to bind to its receptors.
Cortisol binds to both glucocorticoid and mineralocorticoid receptors. The GRs are expressed everywhere and mainly binds cortisol, while the MRs are mainly in tissues dealing with ion balancing, such as the liver, kidneys, and colon. The MRs bind mineralocorticoids, such as aldosterone, less tightly, making it clear why these specialized tissues have the type 2 dehydrogenase that deactivates cortisol, so it does not compete for the MR receptors, but allows aldosterone to drive their function. Important to note is that both receptors are found within the brain, where a lot of the stress response action occurs. To answer the question of how brain GR versus MR receptors respond to stress-induced increases in cortisol, in a 1987 study, researchers induced a stress response in rats and monitored the occupancy of receptors over time. Baseline occupancy was about 30% for GR and 80% for MR, and at peak hormone levels at around 60 minutes, these values rose to 75% for GRs and 97% for MRs, before falling back to near basal levels by 240 minutes. The conclusion was that at basal levels, cortisol functions more through the MR receptor and that GR receptors are much more involved in stress-dependent responses. So, how does glucocorticoid receptor actually work? It is a surprisingly complex process and involves a very large molecular chaperone complex that is required for GR functioning. In this figure, based on a recent cryo-EM structure determination, GR is the yellow protein shown in the middle. So, in the cytoplasm, newly synthesized GR is first bound with the chaperones heat shock protein 70, or HSP70, and HSP40, which hold GR in a partially folded state. Next, ATP hydrolysis causes conformational changes that allows for HOP to bind, whose function is to link HSP70 and an incoming HSP90 protein. This is called the loading complex, in which the yellow GR receptor is loaded into the cleft between the two HSP90 chains in blue. HSP40 and 70 are then released, and two further proteins, P23 and FKBP51, bind to form the mature complex. This complex is ready to bind to cortisol, even though FKBP51 holds GR in a conformation that hinders cortisol binding. This means that when cortisol binds, FKBP51 falls off, and a very closely related protein, FKBP52, binds in its place and promotes transport of the receptor into the nucleus.
The way GR gets to the nucleus is that FKBP52 helps it associate with the motor protein dynein, and the complex, possibly with or without HSP90, is taken along microtubules into the nucleus. At some point, HSP90 is then released and GR dimerizes for binding to DNA binding elements and or other DNA-bound transcription factors. The binding is not long-lived, however, but the GR appears to be fairly rapidly released to release cortisol and cycle off of the DNA and go back into the nucleus, where it's then bound to HSP70 and HSP40 to protect it from aggregation and then to restart the cycle. A contributor to the complexities of cortisol actions is the diversity of ways the glucocorticoid receptor can bind to DNA and modulate gene transcription. As shown here, GR can bind in a variety of ways and produce many different outcomes. The three most common ways are highlighted in red. In its most simple mode, it binds to glucocorticoid response elements, or GREs, as a simple homodimer or heterodimer. Tethering is when other transcription factors or proteins bind the DNA and the receptor then binds to that protein. Finally, composite is then used when the receptor binds as a heterodimer with another transcription factor, and these can lead to the activation or repression or transcription. Overall, studies show that outcomes to glucocorticoid-mediated regulation depends entirely on differences between cell types. So for example, baseline expression of target genes and crotin accessibility, as well as extrinsic factors, including signals from the microenvironment that may affect GR transcriptional regulation and binding abilities. Now that we've looked in detail at some aspects of cortisol synthesis, distribution, and action, we'll turn to look briefly at mechanisms underlying some of the more important systemic impacts of chronic high cortisol levels, and we'll start with its effects on memory.
Stress is well known to impact memory. Memories are dynamic entities that are built in stages, and stress has complex effects on various memory-related processes. This scheme, I emphasize a couple general features of how stress tends to affect memory acting through amygdala and the hippocampus. Briefly, a stressful event leads to the secretion of catecholamines and glucocorticoids, and both of these directly or indirectly stimulate the hippocampus to immediately promote the formation of new memories and hinder the recollection of exciting memories. This explains how even associated with traumatic stress can be seared into one's memory, and then an hour or so later, through the slower genomic action of cortisol, comes a period in which inhibition of the amygdala and the hippocampus leads to the hindering of the formation and storage of new memories that are not related to the stressful event. Looking at two specific landmark studies, one in 1996 looked at cortisol levels and memory performance in healthy adults. First, they showed that expected cortisol levels increased with stress, and then futures showed in the graph circled in red that higher cortisol levels were associated with worse memory recall in terms how many words of a certain kind of person could recall from a list they had looked at earlier. Also gives the well-known importance of hippocampus in memory performance. At 1998 study looked at hippocampal volume in 51 healthy elderly volunteers. They had tracked over 5 years. By comparing a subset with low and decreasing cortisol levels with a subset that had high increasing cortisol levels, they showed that significant prolonged cortisol elevation correlated with smaller hippocampal volume, suggesting a negative impact of chronic stress on the hippocampus and memory formation. Now we'll look at studies showing a couple mechanisms by which cortisol suppresses the immune system.
As a background, antigens stimulate the immune system to produce T helper 1 cells that promote inflammation and activate macrophages to kill pathogens, as well as T helper 2 cells that decrease the activity of pathogen-killing cells and promote tissue repair response. Cortisol blocks the effect of signaling molecules such as interleukin-1 and interleukin-12 that promote Th1 cell differentiation. Thus, elevated cortisol levels due to chronic stress hinder Th1 cell differentiation and create an imbalance, favoring Th2 cells over Th1 cells. In certain acute circumstances, this effect can be beneficial, such as a cortisone shot in the joint of an athlete to reduce inflammation and pain. However, a long-term suppression of the inflammatory response increases the likelihood of illness. The mechanism of IL-1 suppression is a glucocorticoid-mediated blockage of the transcription factor NFKB via changes in the protein P65. The first figure shows how the addiction of dexamethasone, a cortisol agonist, severely reduced cytosolic levels of P65. The second figure indicates the expression of additional P65 limits the inhibition of NFKB signaling by dexamethasone. The model they proposed was that P65 promotes innate immunity by helping remove I-kappa B from NFKB and allowing NFKB to translocate to the nucleus and promote immune response-related transcription. In contrast, the mechanism of IL-12 suppression was determined to be cytosolic cortisol binding to the transcription factor STAT4 and preventing its phosphorylation, as shown in this gel, thus blocking its dimerization and translocation to the nucleus and promoting transcription of interferon gamma. Next, we will dive into the relationship between chronic stress and sleep.
Stress leading to a sleep debt seems intuitive. Generalized anxiety and PTSD are both strongly correlated with insomnia, with anxiolytic medications often being prescribed for insomnia patients. And as seen in this 2001 study, those with insomnia tend to have increased cortisol levels. However, the specific relationships between stress and sleep are more nuanced. For example, one study showed that mice subjected to chronic social defeat stress actually had increased REM sleep. How might this happen? Well, other studies have found that chronic restraint stress results in an increased mouse serotonin production, which, as a happiness chemical, is known to act through the dorsal raphe nucleus to increase sleepiness. This is a part of why the first symptom of major depressive disorder is often insomnia. You might also wonder why hair turns white from a variety of stressors. A 2020 study gave a surprising answer. It showed that the release of noradrenaline due to hyperactivation of the sympathetic nervous system led to the rapid proliferation and differentiation of melanocyte stem cells, leaving no stem cells available to generate melanocytes. Without melanocytes to produce melanin, gray hairs begin to appear. As evidence that this was mediated by noradrenaline, that is, through beta-2 adrenergic receptors, mice with these receptors knocked out, as shown here, did not turn gray in response to chronic stressors. And on the related topic of hair loss, a year later, using mice with their adrenaline glands removed, the same group showed that systemic corticosterone blocks hair regrowth quite dramatically. They further showed that the corticosterone effect is due to the suppressed expression of a gene needed for the regeneration of hair follicle cells, and confirmed it by showing that its overexpression restored hair growth.
Bruce and Keeman reported in 1968 that the hippocampus accumulated cortisol and championed the view that the brain accumulates negative stress-induced changes in adulthood as a result of something he called allostatic load. This is an important concept related to the negative health impacts of stress. First, what is allostasis? It means stability through change, and it was proposed to reflect the reality of life processes better than homeostasis. Because in reality, as illustrated here by the ups and downs of arterial blood pressure, things tend not to be held constant, as the word homeostasis implies, but stably adopt different set point values depending on circumstances. As many changes are experienced, or the body inefficiently manages changes, the concept of allostatic load comes into play. Broadly defined, it is the wear and tear on the body that results from too much stress. A related second theory of McEwen and colleagues is the glucocorticoid cascade hypothesis for aging. The idea is that the more lifetime experiences people have with elevated glucocorticoids, the more likely they are to experience negative metabolic events that coincide with high glucocorticoid levels. One such outcome is a positive feedback loop shown here, in which high glucocorticoids lead to damage to the hippocampus, rendering its HPA axis regulation progressively less effective, leading to ever-increasing glucocorticoid levels. Now we'll transition to consider genetic, epigenetic, and microbiome-related influences on stress and resilience.
In a landmark study from 2008, some variants of the FKBP51 gene were shown to be risk factors for PTSD from childhood trauma. Shown here are 4 resilient and 8 risk alleles, with the risk alleles shown to be associated with increased FKBP51 expression, which would be the expected decrease in cortisol binding to its receptor, leading to GR resistance. In these adults, GR function was tested by the ability of dexamethasone to inhibit cortisol release through the known feedback inhibition of CRH and ACTH release. What they saw was, as shown here, with the individuals with the risk alleles and no PSD showed GR resistance as expected. In contrast, those with PTSD showed GR hypersensitivity, as shown by the much lower cortisol levels after dexamethasone treatment. The authors proposed that the risk alleles enhance the effects of acutely released cortisol following childhood abuse to alter FKBP5 expression, leading to maladaptive changes in GR activity. In 2007, the impacts of the stress response were studied for 5 common genetic variants of the human glucocorticoid receptor. After the Trier social stress test, the 9-beta AG variant stood out as having a higher ACTH response in men, but not women. Also, using dexamethasone suppression of ACTH and cortisol release, it was again the 9-beta AG variant that had unusually high ACTH levels, again only in men. This is an intriguing observation of a possibly highly sex-specific effect of this variant on GC receptor activity. Next, we'll look at some of the epigenetic impacts of stress.
So, lifetime experiences can cause long changes in gene expression without altering the DNA sequence, and effect called epigenetics. With regards to stress, I'll cover a couple of landmark studies showing that DNA mutilation of stress-related genes impact behavior and stress resilience. The studies on the next slide show how epigenetic impacts childhood nurture. Prior to this study, it was proposed that mother-pup interaction impact the stress resilience of and the maternal caregiving by grown pups. This study showed that adults that have been cared for by high-linking and grooming mothers, as seen here, showed markedly reduced plasma ACTH and corticosterone in response to restrained stress. Next, in seeking what underlines the difference in resilience, they found highly increased hippocampus glucocorticoid receptor mRNA levels in the adults who had been well nurtured as pups, which could explain their increased stress resilience. The next question then became how this increased glucocorticoid receptor expression came about. So after some few years, the same group later showed that maternal care caused specific epigenetic changes in pups' glucocorticoid receptor gene promoter region, and this could explain the increase in transcription. Specifically, the cytosine methylation levels in offspring of low nurture mothers, as shown here in black, is higher across the whole region relative to offspring of high nurture mothers. Further on, immunoprecipitation analysis shown that the higher methylation leads to a decreased binding of the transcription factor. And next, they showed that the changes are due to nurturing and not genetics by cross-fostering the offspring of high-care mothers raised by cross-fostering offspring of low-care mothers raised by high-care mothers, shown here as LH, showed a drastic decrease in methylation level. Past offspring of high-care mothers raised with low care, as designated by HL, had a higher methylation level. Further experiment I want to describe shown that it is the high care that causes a decrease in methylation rather than the low care that causes an increase in methylation. And the changes happen within just a week of birth. With this finding, it is easy to understand why this work connecting early nurturing with adult stress resilience and behavior had been cited nearly 7,000 times. Next, we will look at 3 studies showing how the gut microbiome can influence our stress response.
The communication between the gut and brain is bidirectional and involves many pathways. Our examples today involve the most studied HPA axis and vagus nerve routes of communication. Suda and colleagues compared the HPA response to restrained stress of germ-free mice having no microbiome and of mice with a normal functional microbiota. Remarkably, the plasma ACTH and corticosterone increases in response to stress were much higher in germ-free mice. They further showed that colonizing the GI tracts of germ-free mice with Bifidobacterium infantis, shown in green, dampened the HPA stress response, whereas enteropathogenic E. coli, shown in yellow, increased it further. Very clearly, the microbiome matters a lot.
In another landmark study, researchers asked how changes in the composition of the gut microbiome of mice caused by a 7-day antibiotic treatment impacted their stress response. As seen here, the antibiotic treatments dramatically changed the microbiome composition. The treated and control mice were then placed in a box which had a dark room and a light room, and in some cases, obstacles to assess their stress response. The idea is that the more stressed the mice are, the less time they will spend in the light room. Researchers also measured the brain-derived neurotropic factor levels in the amygdala and the hippocampus of these mice. Notably, the antibiotic-treated mice were less hesitant in completing the step-down latency obstacle test and made more entries and spent more time in the lit-up area. They also had much lower brain-derived neurotropic factor levels in the amygdala and higher levels in the hippocampus. 2 weeks after the antibiotic treatment, the behavior in the microbial-fed mice were returned to that of the control group. The control showed that the changes were due to the microbiome rather than any other factor, which powerfully shows that the gut microbiome influences how stress and anxiety affect us.
A third landmark paper looked at the impact of feeding mice with Lactobacillus rhamnosus, shown here as JB1. The treatment reduced stress-related behaviors in both elevated plasmase and forced swim tests, as well as stress-induced corticosterone response. Furthermore, the treatment also induced brain region-dependent alternations in GABA receptor mRNA expression. It was increased in the cortical regions and decreased in the amygdala. None of these effects were found in mice in which vagus nerve was removed, identifying the vagus as a major communication pathway between the gut bacteria and the brain. Now let's turn to studies of some approaches to alleviate stress.
As a reminder, for psychological stress, it's not only the event that matters, but whether people interpret that event as a threat or a challenge, and how they assess their ability to cope with it. This simple diagram, adapted from Gab and colleagues, shows that when a stressor is appraised as a threat, it stimulates HPA axis activity, increasing cortisol levels, which we've seen before is a form of stress. It further shows that we may be able to decrease stress through a positive reappraisal, or reframing of a stressor as more of a challenge than threat. We will discuss 2 categories of interventions to help our appraisal. First, we'll talk about benzodiazepines and how they alter our brain appraisal of stressors, and then we'll discuss behavioral approaches, including meditation, gratitude, guided imagery and music, deep breathing, laughter, and exercise. While there are many other interventions, including positive socialization, healthy diet, time outdoors, art, and more, we hope this will give you some science-based encouragement to proactively manage your stress with a toolset of healthy mechanisms.
Benzodiazepines were one of the first anti-anxiety medications to be widely used. Brought to the market in 1960, they became the most prescribed drug in the world by the mid-1970s. Two recognizable ones are Xanax and Valium. Benzodiazepines are now known to act upon GABA receptors in the brain by allosterically activating them. Recall that GABA receptors are chloride ion channels, and as seen in this plot of current flowing through the channel, benzodiazepines do nothing alone but increase the ion flow at a given concentration of GABA, leading to greater desensitization of the neuron. As seen here, the GABA receptor has 5 related subunits, with the common receptor form containing 2 alpha, 2 beta, and 1 gamma subunit. But it's not as simple as that. Each of the subunits have multiple isoforms. There's 6 alpha, 3 gamma, and 3 beta, which can combine to form a huge variety of specialized versions of the GABA receptor, many of which occur and are heterogeneously distributed throughout the brain. Importantly, benzodiazepine binding is alpha form dependent, and a specific histidine to arginine mutation present in the alpha 4 and alpha 6 isoforms abolishes binding to benzodiazepines. Researchers use this information in their hunt to determine how benzodiazepines work. By making this point mutation in the alpha 1 and alpha 5 subunits, they determine those are not responsible for the anxiolytic effects of benzodiazepines. But what about the alpha 2 and alpha 3 subunits? In 2000, a landmark paper showed that only alpha 2 containing receptors are responsible for the anxiolytic effects of benzodiazepines. Using histidine to arginine point mutations in the alpha 2 and alpha 3 subunits of mouse GABA receptors and a radio-labeled benzodiazepine analog that combined both histidine and arginine containing versions, they did brain imaging. After Valium was used to compete out binding by the radio-labeled ligand, only arginine-containing receptors should show up. The areas lit in both the wild-type and mutated images identify alpha 4 and alpha 6 containing receptors. Areas only lit in the mutant brain contain the subunit under study. Excitingly, the alpha 2 subunit is localized to the hippocampus and hypothalamus, 2 areas that play key roles in stress. Next, they performed a light-dark choice test with the mutated mice. For wild-type mice, Valium strongly increases the time the mice spent in the lit area, as expected. The alpha 3 mutants still respond to Valium, but in the alpha 2 mutant mice, the anxiolytic effects of Valium are abolished. Thus, alpha 2 containing GABA receptors are the anxiolytic target of benzodiazepines. While benzodiazepines are no longer widely prescribed due to their addictive properties, they are still used in situations of debilitating distress. Continued research into the GABA receptor, like this cryo-EM structure, is paving the way for finding new pharmaceutical treatments, as well as understanding the actions of treatments such as selective serotonin reuptake inhibitors. Now we'll move on to talk about behavioral interventions.
Mindfulness meditation is a practice of Buddhism characterized by present-centered awareness, and it's become widespread for stress relief. Garland and colleagues proposed in 2009 that stress reduction by mindfulness meditation is based on broadening one's attention, which improves positive reappraisal, resulting in changing the primary appraisal of environmental stressors from threat to challenge. Studies that contributed to that model include a landmark study in which novices completed an 8-week mindfulness course and were then compared to experienced meditators in the attention network test, ultimately finding that experienced meditators have significantly improved attention. In another well-cited contributing study of 61 college students, a correlation was found between mindfulness ability and the ability to positively reappraise a situation. In 2012, Brand and colleagues asked how mindfulness meditation affects cortisol by comparing the daily cortisol levels of 9 long-term meditators. Cortisol was inversely correlated with the self-reported experience level of these meditators after controlling for age. These findings powerfully support that stress levels are truly decreased by mindfulness. Gratitude is a behavior or feeling characterized by, quote, recognition or appreciation of an altruistic gift. And in a landmark study, Emmons and McCullough assessed the effect of gratitude on positive affect. Undergraduates were placed into 3 groups: a gratitude condition in which participants wrote down up to 5 things they were grateful for weekly for 10 weeks, a hassle condition in which they wrote down up to 5 hassles, and an events condition in which they simply wrote down 5 events that had an impact on them. Based on self-reporting, it was found that those in the gratitude condition showed strongly improved positive affect or mood. So what's going on biologically? Well, 2 years later, another landmark study addressed how happiness correlates with salivary cortisol levels in over 200 participants. They found that after controlling for factors including gender, age, and diurnal cortisol rhythms, there was a strong correlation with people in the highest happiness quintile having the lowest salivary cortisol levels. So it does appear that gratitude improves positive affect and reduces the stress response.
Many of you have probably used music to alleviate stress. Looking into that topic, I found a well-cited 1997 paper on guided imagery and music therapy, or GIM, a psychotherapy method developed by Dr. Helen Bonney and is often referred to as the Bonney method of GIM. In it, specifically selected classical music is hypothesized to evoke imagery for accessing and working through emotional processes. In the study, 28 adults split into GIM and control groups completed the Profile of Mood States Assessment, POMS, and donated 15 cc's of blood before and after 6 biweekly sessions of GIM therapy, and again at a 6-week follow-up. GIM participants reported significant decreases in depression, fatigue, and total mood disturbance, and also significant decreases in cortisol level by the follow-up. Even though formal GIM therapy is administered by licensed therapists, this supports the idea that relaxed music listening can be a great tool for stress relief. An even simpler and more ancient form of stress reduction are deep breathing techniques like those practiced in Qigong and yoga. In a 2017 study of 38 college student volunteers in 2 groups of 19, the question was whether a particular deep breathing technique could reduce stress. Subjects in the experimental group engaged in a 90-minute anti-stress protocol once per week over 10 weeks. It included breathing tied to gentle body motions, vocalizations, and mental imagery, whereas the control group sat 90 minutes each week, not practicing any special treatment. At weeks 0, 5, and 10 of the study, perceived mood and stress were formally evaluated by questionnaires, and a biological stress profile included heart rate and salivary cortisol measurements. As seen in these plots, after the 10-week regimen, the deep breathing group showed significant decreases in perceived psychological stress and in salivary cortisol compared to the control group. Next, let's look at laughter as a fun behavior that we may already subconsciously use to reappraise stress.
A landmark 2002 study of college students asked how humor helped them cope with stressors. Based on several questionnaires, including the perceived stress scale, students were classified as having a high or a low sense of humor. And it was found that the high sense of humor group felt less stressed and less anxious than the low sense of humor group, despite experiencing similar numbers of everyday problems. Another paper that year on laughter ties the psychology studies back to biochemistry. For this study, experimental subjects watched a 60-minute humor video, while the control group engaged in humor-neutral activities. After this, they measured blood samples from an IV catheter for ACTH, cortisol, endorphins, epinephrine, norepinephrine, and DOPAC, which is a catabolite of dopamine. They found that cortisol and DOPAC decreased more rapidly in the experimental group. These results were significant, and the authors concluded that these biochemical changes have implications for the reversal of the neuroendocrine and classical stress hormone response. Finally, you might find interesting a recent review arguing that even simulated laughter, where someone forces themselves to laugh, helps reduce stress. This forced laughter increases dopamine and serotonin, resulting in a more positive attitude and mood, which can lead to positive reappraisals of stressors. Physical activity is another behavior that can help modulate stress, and as noted in our introduction, it's one that many college students use to cope with academic stress. A landmark 1999 review of the benefits of physical activity on mental well-being presents evidence that exercise is effective in reducing acute and chronic stress and treating clinical depression, improving self-perception, global self-esteem, and mood, all of which are factors for a positive mindset surrounding stressors. With respect to stress, 2 cited studies found that acute anxiety was reduced by both single bouts of exercise and a regular exercise regime, and chronic anxiety was reduced by a regular exercise regime. More recently, a 2014 study asked how regular exercise affects emotional resilience to acute stress. Subjects who did and did not regularly exercise were subjected to the TRIR social stress test, and heart rate, blood pressure, cortisol, and self-reported mood were measured before and at repeated times after the stressor. Regular exercisers had a lower heart rate at rest than non-exercisers, and also heart rates that recovered more quickly from the stress. This demonstrates a buffer that exercising provides against stresses. This makes sense considering a third study by Hackney. In this study, exercisers and non-exercisers were subjected to the MAT stress reactivity protocol, which is a stress test that combines a mental, a physical, and a psychosocial stressor. They found that while this activated the stress response in both groups, exercisers responded better and had lower cortisol levels than their inactive counterparts. Hackney concluded that exercise allows the neuroendocrine system to respond and prepare the body for anticipated needs and stressors, both in the present and future. Regular exercise allows for the adaptation and accommodation within the neuroendocrine system so that future stress responses are reduced. While these behaviors we've explored are all great ways to combat stress, our beliefs about stress are also critical and can shape the outcome of a stressor.
Stress mindset refers to whether someone believes that stress enhances or hinders their ability to perform well. In 2017, researchers showed that brief informational videos could successfully manipulate participants' beliefs about stress being either enhancing or debilitating. Then the researchers tested participants with both challenging and threatening social stress tests and found that a stress-enhancing mindset had beneficial effects on cognitive flexibility and mood, independent of stress appraisal. In a 2010 study, researchers had participants read a short paragraph reinforcing that stress enhances focus before taking their practice graduate record examination, and they followed up after they took the real GRE. Those who read the paragraph had substantially improved GRE math scores over those who hadn't. To pass on some of this benefit to you, the paragraph included some of the following. Recent research suggests that arousal can help performance. If you find yourself feeling anxious, simply remind yourself that your arousal could be helping you do well.
So you may consider our ambition to take on this incredibly broad topic of stress as naive and foolish. And I think that we'd all say that you are right. However, we've learned so many valuable things along the way. First, we'd like to know that collaborative study of highly cited landmark papers built great understanding of complex stress systems. Every level of our physiology is stress involved. Judicious investigation of literature provided quite a rich perspective of this nuanced topic. While investigating, we've seen many, many ways to summarize stress. We've come away with a few take-home messages which help us think about stress and resilience. First, how we perceive and appraise stress is fundamental to the complex biochemical responses that happen in our bodies. Evidence points to meaningful differences between a stress mindset of taking on a challenge and viewing a stressor as a threat. Also, evidence shows numerous measures can effectively increase resilience to stress, thus improving outcomes of both health and mood. These include exercise, meditation, medication, and so many more. Lastly, systems of stress involve a lot of very cool and very complex biochemistry. From differential transcription to effects on memory, stress is a fascinating topic.
Finally, we're grateful to the BV department for the environment and training it's provided, and to the many researchers who are helping provide society with strategies and tools for improving wellness through enhancing resilience and limiting stress, and to our family and friends and others who have supported us in getting to where we are. Thanks also to you for your attention. And now we'd be glad to take questions.